So today's topic is rolling circular replication and inhibitors of replication. First, we will discuss what is DNA replication. So DNA replication means it is a process by which the DNA will be producing multiple copies. That is, they will be producing identical daughter DNA strands from their parental strand. So this will be taking place in a semi-conservative process. And also the biological polymerization which proceed in the sequence is of initiation, elongation and termination. There are 3 steps, and also there are many enzymes involved. Most of the enzymes or the main enzyme is DNA polymerase enzyme. Then rolling circle replication. So the rolling circle replication means this is a process which a circular DNA or RNA molecules will be replicated only in one direction. So most of this rolling circle replication will be mostly seen in virus genome and also in plasmids. So this rolling circle replication will be producing multiple copies of a single circular template. There are many enzymes involved in this. One is RUP-A protein, DNA polymerase 3, helicase enzyme, helicase enzyme. So RUP-A protein means which is made up of 2 dimers. So they are having 2 active tyrosine group also, which helps in the smooth functioning of the rolling circle replication. And second enzyme is DNA polymerase 3, which means this polymerase enzyme means that will be adding new nucleotides to the strand, which means it will helps in the elongation of the DNA. Helicase enzyme which helps to unwind the DNA, and ligase enzyme that helps to join the gaps formed in the DNA. Next is the rolling circle mechanism. So what happens is the double-stranded circular DNA will make a cut on that DNA. So nick is made in the strand and the 5' end will be displaced, and polymerization at the 3' end, that means adding of new nucleotides, and at last attachment of the replisomes and formation of Okazaki fragments. So the first step is initiation. So for initiation, what happens is the double-stranded circular DNA is there. So the rolling circle replication is initiated by an initiator protein, that is RUP protein. So there are 2 sites for this double-stranded DNA. One is DSO and SSO. DSO means double-stranded origin and SSO means single-stranded origin. So the nick will be produced in the DSO site, that is double-stranded origin. Second is elongation. So what really happens is the initiator protein will produce a nick, and this initiator protein will be binding to the 5' phosphate of the nick strand, and the 3' hydroxyl end of the next strand is elongated by DNA polymerase. So this 3' hydroxyl end of the next strand get elongated by using template strand. The template strand which is used as the unnicked strand. The unnicked strand is used as a template strand so that the nicked strands will be producing new nucleotide. The 5' phosphate nick strand is displayed. As a result, the 5' phosphate nick strand will be displaced by the helicase enzyme. Termination means the linear DNA molecules that which is cleaved from the circle will be producing another double-stranded circular DNA molecule and a single-stranded linear DNA molecules. So the cycle will be repeated to make many number of single-stranded copies. The next is inhibitors of replication. So inhibition or inhibitors of replication means it will prevent the cell division. There are many substances which can be used as inhibitors of replications, such as dauromycin and adriamycin. So these both are synthetic chemotherapeutic agent, that is, they are the inhibitors of both DNA replication and transcription in prokaryotes. So these are presumably acting as interfering with the passage of both DNA and RNA polymerase. They will interfere the passage of the DNA and RNA polymerase enzyme. So they are having planar aromatic ring system which gets intercalated between GC pairs of the double helical structure. They also prevent replication and transcription. The second one is adriamycin. Otherwise, these are called as doxorubicin. So this doxorubicin is cytotoxic anthracycline antibiotic, And this deoxyribosin consists of a naphtha as an quinone nuclease linked through a glycosidic bond at ring atom 7 to an amino group or amino sugar daunosamine. So which may inhibit the animalian topoisomerase, and also it is used as a cancer drug. Next is ethidium bromide and proflavin. So these both will be inhibiting the replication and transcription by intercalation. Also, these are a fluorescent compound. It is also aromatic. So the main portion of the molecules is tricyclic structure with alanine group on either side of pyridine. That is this dibenzopyridine structure, which is also known as phenanthridine. Next is actinomycin D. So this is an antibiotic which is produced by streptomycin and which inhibit the replication and transcription. So this also acts as a intercalating agent. This actinomycin D has two identical pentapeptide which have unusual composition of D-valine and sarcosine, which stabilizes this intercalating interaction. Next is quinolines. These are antibiotic. They also interfere DNA replication by inhibiting topoisomerase, DNA gyrase, and ciprofloxacin. They also interfere with DNA breakage and rejoining process. These are my references.